Welcome to the deep dive. Today we're diving into something pretty fascinating, right at the intersection of AI and biology.
Yeah, it's a really exciting space.
We're looking at how some really advanced AI is, well, changing the game in life sciences research. Think revolutionizing medicine, maybe even extending our healthy years.
Absolutely. The pace is just incredible right now.
So our main source for this deep dive is a really key publication from OpenAI. It details their work with a company called Reprograms. And our mission really is to unpack how this unique, specialized AI model, call it GPT-4B micro, is making these huge leaps in areas like stem cell reprogramming and maybe even cellular rejuvenation.
That's a big deal.
Yeah, and I think the core thing we'll keep coming back to is this idea of building AI that's, fine-tuned, really specialized for these specific scientific jobs.
Right. It's not just about bigger is better. It's about smarter, more focused tools. And what's fascinating here is how quickly these advancements are moving from, theory to actual results. This isn't just lab stuff. It holds real promise for therapies, for understanding aging, the potential for medicine, for longevity. It's truly immense.
It really is. And central to a lot of this regenerative biology work are these things called the Yamanaka factors.
Ah, yes. OCT4, SOX2, KLF4, and MYC.
That's them. The proteins that can create induced pluripotent stem cells, iPSCs. Nobel Prize-winning stuff, right?
Landmark discovery, yeah.
And the potential uses for these iPSCs are, well, frankly, enormous. Imagine using cells to fight blindness, reverse diabetes.
So infertility, even grow organs for transplants, address those critical shortages.
Exactly. These cells could be the key. But there's always a but, isn't there?
Yeah, usually.
Despite all that promise, using these Yamanaka factors efficiently has always been a massive problem. The reprogramming efficiency is just notoriously poor.
Yeah, really low.
We're talking less than, 0.1% of cells actually converting?
Often, yeah. And it can take 3 weeks, sometimes even more.
3 weeks. And it gets even worse if you're dealing with older cells or disease cells, right? The efficiency just tanks. It's a huge bottleneck.
It absolutely is. And if you look at the bigger picture, it really highlights a fundamental challenge in biological engineering.
So why is it so hard?
Well, it comes down to the numbers. The sheer, almost unimaginable number of ways these proteins could possibly be arranged.
Like permutations.
Exactly. Take SOX2, it has 317 amino acids. KLF4 has 513. The number of possible sequence variants is just astronomically high. Think 10 to the power of 1,000.
10 with 1,000 zeros.
Yeah.
I can't even picture that.
Nobody can, really. It's more possibilities than atoms in the universe, by orders of magnitude. So traditional methods like these directed evolution screens, they can only explore a tiny, tiny fraction of that massive design space.
Right. It's like looking for a specific grain of sand on all the beaches in the world, blindfolded.
Pretty much. A good analogy.
So you mentioned earlier 15 years of work on just tweaking LSX proteins, right? And after all that, the best variants only differed by maybe 5 amino acids.
Just 5 residues, yeah. That really shows you the scale of the problem we're facing with traditional approaches.
Okay, so that sets the stage perfectly. How on earth does an AI, even a smart one, tackle that kind of impossible haystack?
Well, this is where GPT-4B micro comes in.
The star of the show.
Indeed. And that micro name is interesting because its specialized, smaller nature is actually key to its power here.
So it's not just a shrunk-down GPT-4, it's tailored.
Precisely. It speaks to that idea you mentioned for these really tough scientific nuts to crack. A focused, fine-tuned AI might be much more effective than a massive generalist model. This thing was built for protein engineering.
Got it. A specialist. So what's special about its training?
Okay, so several things. First, it was initialized from a scaled-down version of GPT-4o.
OpenAI's big multimodal model, right? Text, images, audio.
The very same. So GPT-4B Micro got a head start, inheriting some of that powerful base knowledge and reasoning capability. But then it got further intensive training specifically on a huge data set of protein sequences.
All right, makes sense.
But here's a crucial difference from many other protein language models. It wasn't just sequences. It also learned from biological text, descriptions, and tokenized 3D structured data.
So it understands the protein's shape, not just the sequence.
In a way, yes. It breaks down those complex 3D shapes into digital pieces it can process alongside the sequence and text. And there's more.
Right.
A lot of the training data was enriched. They added extra context, like textual descriptions of function, data on how similar proteins evolve together in other species. That's co-evolutionary data, which gives clues about function,
Okay.
and even information about which proteins interact with each other in the cell. It's building a much richer picture of the protein and its biological environment.
That sounds incredibly important, especially for proteins like the Yamanaka factors. Don't they have regions that are kind of floppy, intrinsically disordered?
They do, yes. Intrinsically disordered regions, or IDRs. Their function relies on flexibility and interacting with many partners, not locking into one single rigid shape.
So a model just focused on a single structure would miss a lot.
It likely would. GPT-4b micros training, with all that sequence, structure, text, and interaction context, seems much better equipped to handle that complexity. It's kind of a game changer for understanding these dynamic proteins.
Right. And you mentioned something about context length.
Yes, that's another technical leap. Because they trained it on this enriched data, the effective context window, how much information it can consider at once, became much larger. They could use prompts up to 64,000 tokens during inference.
64,000. That's huge for a protein model, isn't it? Common for text, maybe, but proteins.
Exactly. It's pretty much unprecedented for protein sequence models, which naturally leads to the question, okay, impressive tech, but does that massive context actually help in the real world?
Does it translate? That's always the billion-dollar question with AI, isn't it?
Mm-hmm.
Theory is one thing, practice.
Right. So OpenAI saw promising signs during development. The model followed similar scaling laws to language models, bigger models, more data, predictable improvements.
Okay, good sign.
But, testing AI protein designs just on a computer in silico only tells you so much. The real proof is in the pudding, or in this case, the petri dish.
The wet lab results.
The wet lab. That's where the collaboration with Retro Biosciences becomes absolutely crucial.
Right, their applied AI team.
Right.
So they took GPT-4b micros designs.
And tested them. Retro built this dedicated screening platform using human fibroblast cells, standard skin, and connective tissue cells to see if the AI-designed SOX2 and KLF4 proteins actually worked better.
Okay, moment of truth. And the results for these retro-SOX sequences.
Pretty stunning, actually. Get this.
Wow.
Over 30% of the AI suggestions worked better than the natural wild-type SOX2 at turning on those key stem cell genes.
30%. That sounds really high for this kind of thing. What's typical?
Usually you'd be happy with a hit rate below 10% in traditional screens, sometimes much lower. So yeah, 30% is a massive improvement.
Wow. And these weren't just tiny tweaks either, right? You mentioned they were quite different from the original protein.
Hugely different. On average, the successful variants had changes in more than 100 amino acids.
100. Compared to the 5 from 15 years of traditional work.
Exactly. It's a totally different scale of exploration, far beyond what you could rationally design or find through random mutation and selection alone.
That's incredible. Okay, what about KLF4? You said that one was even trickier.
Much trickier. Remember, previous attempts by experts making single amino acid changes had only found one improved version out of 19 tries. Very low success rate.
Right, a tough nut to crack.
Yeah.
So how did GPT-4b micro do with retro KLF?
Even better, remarkably, nearly a 50% hit rate.
50, half of them worked better.
Half of them. And 14 of those AI-generated KLS variants were actually superior to the best cocktails they'd found using the improved retro SOX.
So the AI just kept getting better.
Yeah.
But the real kicker is when they combine them, isn't it? The best SOX with the best KLF.
That's where it got truly dramatic. Using the top retro SOX and retro KLF variants together, they saw a greater than 50-fold increase in the expression of those key stem cell reprogramming markers compared to the standard factors.
50 times higher.
50 times. And not just higher expression, those important late-stage markers, the ones that really show reprogramming is complete, they appeared several days earlier, too.
So faster and much more efficient. That's not just tweaking, that's dot transformative.
It really feels like a quantum leap. And critically, these weren't just flukes in one experiment.
Ah, the robustness check. Always important.
Absolutely essential if you're thinking about therapies down the line. They replicated the results using cells from different people, different types of cells, even connective tissue stem cells from middle-aged donors. And different delivery methods, too. They tried using mRNA to deliver the factors instead of viruses, and it still worked. And the final iPSC lines they created, fully pluripotent, genetically stable, ticked all the boxes.
Wow. So it really works reliably That's huge.
It's very strong validation. It shows the AI isn't just making random changes that happen to work sometimes. It's genuinely designing improved, functional, stable biological components.
Okay, so better stem cells, faster, amazing. But then they looked at something else too, right? Something about rejuvenation.
That's right. They didn't just stop at making iPSCs more efficiently. They wondered if these souped-up factors could also be better at tackling aspects of cellular aging.
And they focused on DNA damage.
Exactly. DNA damage is seen as a core driver, a canonical hallmark of the aging process. And there was already some evidence that the original Yamanaka factors could help erase some DNA damage markers, even without fully turning the cells back into stem cells.
Sort of a partial rejuvenation effect.
Kind of, yeah. So the question became, could these new AI-designed RetroS and RetroKLF variants do an even better job at cleaning up that damage?
Makes sense. So how do they test that?
They used a standard DNA damage assay. They treated cells with a drug, doxorubicin, which causes double-strand breaks in DNA, a really serious form of damage. Then they looked for a marker, OH2AX, which lights up when these breaks occur.
Okay, so more light means more damage. What did they find?
The cells treated with the RetroSOXKLF cocktail showed visibly less of that H2AX signal compared to cells treated with the standard factors or a control group.
Less damage signal. So the AI factors were helping repair the DNA or preventing the damage accumulation better.
That's the implication. It strongly suggests these engineered variants are more effective at reducing this key type of DNA damage than the original Yamanaka factors. They're actually ameliorating a core hallmark of cellular aging more effectively.
Wow. So they're not just better at reprogramming, they might actually be better at repair and rejuvenation too.
It certainly points in that direction. It really opens up intriguing possibilities for future therapies, doesn't it? Especially for age-related conditions where DNA damage plays a big role.
Definitely. So let's try and wrap our heads around this. What's the big picture here? We've got these incredible results, super high hit rates in the screens.
Right, over 30%, nearly 50%.
Really deep changes to the proteins, like over 100 amino acids different.
Unprecedented scale of change.
Making the whole process much faster, markers appearing days early.
Instant acceleration.
Robust results across different cells and methods, and now this potential for enhanced DNA damage repair. It's quite the list.
It really is, and I think it powerfully demonstrates OpenAI's core idea here. AI can significantly speed up innovation in life sciences. This project is a fantastic example of how a domain-specific model, GPT-4b Micro, designed for a particular job, can yield breakthrough results on a really focused, really hard scientific problem.
It really pushes back against the idea that you just need one giant AI for everything, doesn't it? Sometimes a specialist tool is what you need.
Exactly. It's about the right tool for the job. Smart, targeted models.
There's a great quote from Boris Power at OpenAI, who handles these research partnerships. He said something like, when researchers bring deep domain insight to our language model tooling, problems that once took years can shift in days.
Shift in days, that captures it perfectly. That's the kind of acceleration we're seeing.
And it hammers home that point about fine-tuning, doesn't it?
Yeah.
Creating these specialized versions of big models for specific, complex scientific tasks seems like a really powerful strategy. That might be where the biggest gains are hiding.
I think that's right. That combination of broad capability from the base model plus deep specialization from fine-tuning is incredibly potent.
So we've been on quite a journey here with GPT-4b Micro. Designing these supercharged proteins, boosting stem cell creation, enhancing DNA repair. The implications for cellular rejuvenation for new therapies are just huge.
And you feel like this is really just scratching the surface, for AI in biology. This work highlights how fast things can move when you pair these targeted AI tools with real human biological expertise. That synergy is key.
Absolutely. The human insight guiding the AI tool. So this deep dive showed us how problems taking years can now shift in days. It really makes you think, doesn't it? What other huge scientific hurdles, maybe completely outside protein engineering, are just waiting for the right specialized AI model?
What else could be accelerated like this?
Exactly. What breakthroughs are sitting there, maybe just around the corner, just needing that perfectly fine-tuned AI tool to unlock them? Food for thought.
Definitely.
Thank you for joining us on this deep dive.